Dear student, I am Dr. Prakash Mungli, Professor of Biochemistry. So in this video, I will be going over catabolism of carbon skeletons of amino acids. So in my previous lecture, I have explained what happened to the amino group nitrogen, whereas here I will be going over the catabolism of carbon skeleton of amino acids. In that, I will go over amino acids that form pyruvate. Let's get into the details. After transamination reaction, meaning whenever the alpha carbon amino group is transaminated means transferred from an amino acid to other alpha keto acid. At that time, the carbon skeleton that remains, so what will happen to that carbon skeleton? So we are going to look into that. That is what is the catabolism of carbon skeletons of amino acid. Let me write down for your easier understanding what actually I mean by catabolism of carbon skeletons of amino acids. Okay, so here I have written the structure of alanine. This is the structure of alanine, where you see there is an alpha carbon in the center, primary carboxyl group, primary amino group, and the side chain of alanine, as you know, is methyl group. So when an amino group is transaminated, that means when this amino group is removed, so when it is taken out, so by transamination reaction, we have already seen this, what is the meaning of transamination and all the important points and everything about transamination. So we have seen that in our previous lecture. Now let's see what will happen to the carbon skeleton that is remaining once this amino group is transaminated and taken out. So I am going to remove that part now. So amino group is not there now, it is removed. Now the remaining carbon skeleton is, it will be released as pyruvate. Now this is pyruvate. After the transamination reaction, the carbon skeleton that is remaining from alanine is pyruvate. So what will happen to the pyruvate? That is what is the catabolism of carbon skeletons of amino acids. Here it is alanine. So what will happen to the carbon skeleton of alanine, which is now released as pyruvate? So what will happen to the pyruvate? Pyruvate can have two fates. Pyruvate, it can be getting into the mitochondria and be converted into acetyl-CoA. Pyruvate can be converted to acetyl-CoA and it can go into TCA cycle. That's one of the fate of pyruvate, right? Completely oxidized, or that is used for energy purpose. Or pyruvate, if the person is in fasting condition, pyruvate can go into glucose formation. That is done by gluconeogenesis. And this is what is the catabolism of carbon skeleton of pyruvate. And like this, for every amino acids, we are going to see what is the fate of carbon skeleton of each of these amino acids after they undergo transamination or direct deamination process. That is what is the purpose of today's lecture, to understand what will be the fate of the carbon skeleton of an amino acid after the amino group is removed. Okay, that's the basic purpose of today's lecture. That is, we need to know what happens to the carbon skeleton now. Let's get into the details. After transamination, carbon skeleton of amino acid forms one of the following. That is, either it will form pyruvate, just now what I have explained, remaining carbon skeleton can form pyruvate, or remaining carbon skeleton can form intermediates of TCA cycle, and that can be like alpha ketoglutarate, or it can be succinyl-CoA, or it can be fumarate, can be oxaloacetate. So any of these can form from the carbon skeleton of amino acids. Or they can make acetyl-CoA directly, or they can make acetoacetyl-CoA. So these are the fates. Now what will happen to the pyruvate, what will happen to the intermediates of TCA cycle, what will happen to acetyl-CoA, what will happen to acetoacetyl-CoA, we'll look into that. Based on what will be the fate of pyruvate, intermediates of TCA cycle, acetyl-CoA, acetoacetyl-CoA, depending on the fates of these end products. So amino acids can be classified into glucogenic amino acids, the ones that will make glucose, or ketogenic amino acids, the ones that will go into ketone bodies, and both glucogenic and ketogenic amino acids. So this will happen. This will be the fate of the intermediates that are made here, pyruvate, TCA cycle intermediates, acetyl-CoA, acetoacetyl-CoA. Let's look at the list of amino acids that will make glucose, the list of amino acids that makes ketone bodies, and the amino acids that make both glucose and ketone bodies. So here is the table that will give you all the names of amino acids that will make glucose, which are called as glucogenic amino acids, which sometimes referred as glycogenic amino acids. Don't get confused, glucogenic or glycogenic. And the other is which are converted to fat like ketogenic amino acids, and then the ones that are converted to both glucose and ketone bodies. So first let's remember what all the ketogenic amino acids, and there are only two of them and easy to remember, leucine and lysine. Leucine and lysine are the ketogenic amino acids, and you can remember as a mnemonic Lucy-Lys. You can make your own mnemonic. And then the both glucogenic and ketogenic amino acids are in that three aromatic amino acids are there: phenylalanine, tyrosine and tryptophan. And then there is one branched chain amino acid, that is isoleucine. So these are both glucogenic and ketogenic amino acids. So rest all of them other than these six, they are all only glucogenic amino acids. So you must remember these. Okay, it is important. So leucine and lysine are ketogenic. Isoleucine, phenylalanine, tyrosine, tryptophan are both glucogenic and ketogenic. Rest all of them are glucogenic. Now let's move on to see the amino acids that form pyruvate. The amino acids that form pyruvate are given in this particular slide, very simplified, easy to remember. Of course, they will go through intermediates, but at the end, we need to know what all the amino acids that form pyruvate and pyruvate going into glucose formation. So that is threonine, which is converted to glycine, and glycine further converted to serine, and serine going into pyruvate. So threonine, glycine and serine making pyruvate. On the other side, cysteine and alanine making a pyruvate. And also note that threonine can also go towards acetaldehyde formation and acetyl-CoA formation. So in that sense, so threonine can also make ketone bodies by giving acetyl-CoA. Okay, so some books will mention threonine also as both glucogenic and ketogenic. Why? Because threonine, by making pyruvate can go into glucose, and threonine by making acetyl-CoA can make ketone. So that's why threonine, some of the books will mention it as both glucogenic and ketogenic, whereas in the table that you have seen here, you can see threonine is mentioned here as only glucogenic. So I just wanted to bring that to your attention, just in case. This is a area where there can be a bit of confusion. So I will have to go according to how the question is being framed. So it can come to this side also, threonine, into both glucogenic and ketogenic. Alright, so there are 5 amino acids here, going by that explanation, which are both glucogenic and ketogenic. So isoleucine and threonine, and then 3 aromatic amino acids, that is phenylalanine, tyrosine, tryptophan. Alright, so I just wanted to give you that area of confusion and that you must be aware of. Now, going by that, as you have seen, alanine, which is making pyruvate, cysteine making pyruvate, threonine, glycine, serine making pyruvate. Let's look into little more details. I'll be going over the high yield points just to make sure that you know little more details how all these amino acids are going into pyruvate. Here is like transamination of alanine. Whenever alanine undergoes transamination, so alanine will give a pyruvate because the amino group of alanine will be transferred to alpha-ketoglutarate in transamination reaction, and the carbon skeleton of alanine will be released as pyruvate, while alpha-ketoglutarate will be converted to glutamate after receiving amino group from alanine. This is a classic transamination reaction. So upon transamination, alanine will be converted to pyruvate. That's how the pyruvate will come from alanine. So alanine is the one that makes pyruvate. Pyruvate is glucogenic. So that's why alanine is glucogenic. Now, dehydration or transamination of serine can make pyruvate, as it is shown here. Serine undergoes dehydration to make a transient intermediate, and then there will be formation of pyruvate, and there will be release of ammonium ion in this process. So serine undergoes a deamination process, and also serine can make 3-hydroxypyruvate, and this 3-hydroxypyruvate further it will make a glycolytic intermediate, that is 3-phosphoglycerate. So that can also form. So that means serine is glucogenic because it will make pyruvate, and also it will make a glycolytic intermediate 3-phosphoglycerate, which can be reversed all the way to glucose in gluconeogenesis. That's how serine makes pyruvate here, and that's how serine is a glucogenic amino acid. Now coming to the cysteine. So how cysteine is converted to pyruvate? Cysteine can be converted to pyruvate by loss of sulfur atom, because you know cysteine is a sulfur containing amino acid, and this sulfur atom which is lost, it will be released as either hydrogen sulfide, that is H2S, or it can be released as sulfite, which will be excreted as sulfate, or it can be stored in the body as phosphoadenosine phosphosulfate, which is the sulfate donor in our cell. Now how it is done? It is shown here in the figure. Cysteine initially form metapyruvic acid and later converted to pyruvate after separation of hydrogen sulfide. That's one way. Other way is cysteine converted into cysteine sulfonate, and that cysteine sulfonate later will be converted to sulfonyl pyruvate, and then the pyruvate is released and sulfite is released, and that sulfite will be converted to sulfate, that is SO4 minus. Now one of the intermediate in this process is cysteine sulfonate, and this cysteine sulfonate it can go towards a taurine formation. Taurine. So cysteine is the one that gives taurine upon its catabolism of carbon skeleton. Now what is the importance of taurine? So you must have studied dietary lipid digestion and absorption. So in the dietary lipids digestion and absorption, there is the requirement of bile acids, and these bile acids are synthesized in the liver, and the primary bile acids that are synthesized in the liver are cholic acid and 𝗑𝐴𝐳 acid. So cholic acid and 𝐼𝐻 acid, these are the primary bile acids that are made in the liver. These two bile acids will undergo conjugation process. So bile acid conjugation, and the bile acid conjugation, one of the conjugating molecules in bile acid conjugation in the liver is a taurine. So bile acids are conjugated with taurine, and conjugation of taurine with bile acids will decrease the pKa of these bile acids from 6 to 2, pKa 2. Thereby, it increases the efficiency of bile acids in the duodenum. So, in my lecture on bile acid synthesis, I have already explained you details how exactly this goes on in the hepatocytes and what is the use of bile acid conjugation. So, in that process, so cholic acid and 𝐼𝐻 acid uses a taurine that is coming from cysteine degradation, and cholic acid becomes taurocholic acid, 𝐼𝐻 acid will become tauroChenodeoxycholic acid. That's an application there, you must remember. Now let's move on to see fate of carbon skeleton of glycine, because glycine is another amino acid that makes a pyruvate. So let's see how that happens. Glycine is oxidized into carbon dioxide, ammonium ion and N5N10 methylene tetrahydrofolate by a glycine cleavage enzyme, which is a reversible reaction. Note that glycine can undergo three fates in our cells. Carbon skeleton of glycine can have one of these three fates that I am explaining now. And this is important, especially in relation to the forms of tetrahydrofolate that we are going to get here, because glycine metabolism is the one that gives a type of tetrahydrofolate which can be used in certain important metabolic processes, like to make in the conversion of DUMP, that is deoxyuridine monophosphate, into DTMP, deoxythioridine monophosphate, in the DNA synthesis, right? So we need to know the source for that is glycine catabolism. So glycine is oxidized into carbon dioxide, ammonium ion and N5N10 methylene tetrahydrofolate by a glycine cleavage enzyme, and shown here. Glycine tetrahydrofolate and NAD plus getting into reaction catalyzed by glycine cleavage enzyme and releasing N5N10 methylene tetrahydrofolate. So the carbon of glycine will be released as means alpha carbon. Alpha carbon of glycine will go into formation of N5N10 methylene tetrahydrofolate. That's where its carbon goes. And the carboxyl carbon released as carbon dioxide, amino group released as ammonium ion, and NAD is reduced into NADHH+. So that's one fate of breakdown of glycine's carbon skeleton and also the amino group released as ammonium ion. Now let's look at the second reaction that can handle carbon skeleton of glycine, and that is glycine can be converted to serine by the transfer of one carbon from N5N10 methylene tetrahydrofolate by serine hydroxymethyl transferase enzyme and making a serine. So that means glycine is converted to serine while N5N10 methylene tetrahydrofolate is converted to just tetrahydrofolate. So that's another possible fate of glycine. So glycine can, carbon skeleton and amino group can be directly released by glycine cleavage enzyme. Second fate is glycine can be converted to serine while N5N10 methylene tetrahydrofolate is simply released as tetrahydrofolate. And the third fate of glycine is glycine can be converted to glyoxylate by glycine oxidase or D-amino acid oxidase, and this happens in the peroxisome. The third reaction that you are seeing here is the peroxisomal reaction, and also the one of the enzyme that is involved in the conversion of glycine into glyoxylate and then into alanine. So that will be done by alanine glyoxylate transaminase enzyme, that is AGT, alanine glyoxylate transaminase, and that enzyme is present in the peroxisome. So like this, the carbon skeleton of glycine undergoes 3 fate. Let me repeat that one more time. It is glycine is oxidized completely by glycine cleavage enzyme, and in that process N5N10 methylene tetrahydrofolate is made. Second fate is glycine is converted to serine by serine hydroxymethyl transferase enzyme. In that process, the carbons of glycine going into formation of serine. And the third is the one that happens in the peroxisomes, where glycine is converted to glyoxylate via glycine oxidase, and or it can be going into D-amino acid oxidase, or it can be oxidized by D-amino acid oxidase. So one of the enzyme involved in the conversion of glycine to glyoxylate in that pathway is alanine glyoxylate transaminase, that is AGT. Deficiency of alanine glyoxylate transaminase enzyme, it will give rise to a primary hyperoxaluria. So we are going to see details of primary hyperoxaluria later. Now, the last one is conversion of threonine into pyruvate. So threonine is converted into glycine and acetaldehyde, and glycine is then metabolized as above in number 1, 2, 3 reaction that we have explained. So this is how glycine either completely broken down into N5N10 methylene tetrahydrofolate, carbon dioxide and ammonium ion, or glycine making serine, and serine further it is converted to the pyruvate that we have seen in the previous slides. That's how glycine goes into formation of pyruvate, and threonine by making glycine making serine going into pyruvate formation, as it is shown here in this slide. You see, threonine to glycine to serine, and serine to pyruvate, or glycine making serine to pyruvate. So that's how all these amino acids will make ultimately pyruvate as the intermediate, and that pyruvate goes into glucose formation. So this is the fate of carbon skeleton of amino acids, and the fate of carbon skeleton of amino acids that makes only pyruvate I am explaining in this lecture. So the purpose of this lecture was to look at the catabolism of carbon skeleton of amino acids that form pyruvate. Just I have covered only the amino acid that makes pyruvate. So that's about it. In my next lecture, I will explain the catabolism of carbon skeleton of other amino acids, how they go in. Thank you.